So the first passage that I will dissect for the biology section of the exam is from MCAT Official Practice Exam 2, passage number 5. And just before I begin dissection, I'm going to start with my major rules of thumb on how to approach a biology passage and how to strategize our way through it and how to answer questions even if we don't necessarily know what is going on. So whenever I read a biology passage, the first time, I mostly don't have 100% idea of what is happening because that's normal. But I want to read fast and answer slow. So instead of taking time to read and try and understand everything, I want to strategize my time and focus on the questions and then go back and try to find answers. Because remember, they're not going to ask you questions about every part of the passage, and the questions and the answer choices will help guide you and help you understand and help you discover what is really going on here. So I've taken the time to read this passage, and you can do this on your own time, but I will start attempting the questions. So the first question asks, the enzyme encoded by the TDH2 gene catalyzes the reversible conversion of what? So I see that TDH2 gene is mentioned here, and it says right here that it encodes glyceraldehyde 3-phosphate dehydrogenase GAPDH. Now, first of all, before I walk into the exam, I should have a very good idea of all of the enzyme classes and what they do. Now, dehydrogenase is an oxidoreductase enzyme, so that means it takes off hydrogen from a molecule and it puts that hydrogen onto something else. Usually it's NAD, but it can be anything. So that's in general what a dehydrogenase does. But for example, I walk into an exam and I don't know that, or I don't remember that. That's perfectly fine, because the other thing about enzymes is that the general rule of thumb is that enzymes are named for the substrate on which they act. So if the enzyme is named glyceraldehyde 3-phosphate, then that means this is the substrate that the enzyme acts on. So right off the bat, I can see that B mentions glyceraldehyde 3-phosphate as the substrate. So I would be very tempted to pick B, but just in case, I will go through the rest of the answers as well. So 3-phosphoglycerate to 1,3-bisphosphoglycerate, this is not a dehydrogenase reaction. Fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate, this is a major step in the glycolysis reaction, and again, this is not a dehydrogenase reaction. 2-phosphoglycerate to 3-phosphoglycerate, here I'm just moving around the phosphate groups, again, not a dehydrogenase reaction. So I'm not removing any hydrogens in A, C, and D, so they cannot be done by a dehydrogenase. And on top of that, the final piece of evidence I have is that glyceraldehyde 3-phosphate is my substrate. So on to the next question. This is a good one, and this is the type of question that is a thinking question, and you can never study enough content to be able to answer a question like this. The question asks, based on figure 1, how do the deletions of the HST3 or the RTT genes affect the lifespan of yeast? So now I'm going to go to figure 1, and I'm going to first take a look at what's happening. So my x-axis is the lifespan, and my y-axis is the percent survival. So whenever I'm reading a figure or an experiment or anything that has to do with research figures or research tables or diagrams or whatever, the first question I need to be asking is what is changing with respect to my normal or my control or my wild type. So always measure change. What's important is what's changing, what is different. That is what will tell me what's happening, because again, if I'm a research scientist, I'm interested in what is changing as a result of my experiment. I don't care about what is the same. And when I talk about change, I'm referring to with respect to normal. So here, my wild type is normal, and I can see that these black triangles are also normal. So they're behaving normally, right? So that means when I remove RTT, remember delta RTT means I'm removing the RTT gene, nothing happens. Everything is normal. Then when I take out the HST3 gene, something happens, not normal. And when I take out both, again, similarly, something happens, not normal. So now let me go and let the answer choices guide me to answer the questions. This little summary that I did was just for understanding, but in an exam, I wouldn't take this much time. I would straight off the bat go to the answer choices and let them just guide me through. A says the deletion of the HST3 gene compensates for the RTT-induced decrease in lifespan. So delta RTT-induced decrease in lifespan, that means that delta RTT induced decrease in lifespan, first of all, I don't think that there is a decrease in lifespan necessarily because it's the same as wild type, but the exam makers might be referring to the natural decrease in lifespan, so I'm not going to really pick at that. But the answer choice is saying that when I delete the HST3 gene, this compensates for that. So when I delete the HST3 gene, does that compensate for what's happening here? No, it doesn't. So again, I can also look at the white triangles. In the white triangles, I have RTT deleted and HST3 deleted. And then in the black triangles, I have just RTT deleted. So there's no compensation. In fact, without just RTT, my cells survive much better. Their lifespan is longer. B says, the deletion of the HST3 gene has no effect on the RTT-induced increase in lifespan. So yes, RTT does actually increase lifespan, but then when I delete HST in this white triangle condition, does that have no effect? No, it does have an effect because I go from black triangle to white triangle. So I'm comparing no RTT versus no RTT and no HST3. Then C says, the deletion of the RTT gene compensates for the HST3 induced decrease in lifespan. So first of all, let me look at HST3. So, okay, when I delete HST3, black circle, I do have a decrease in lifespan, that is correct. But then, on top of that, when I delete RTT as well, does that compensate? White triangle, I take out HST3, but then I also take out RTT. Does that compensate? Does that fix the problem? No, it does not. The problem is still there. And finally, D says the deletion of the RTT gene has no effect on the HST3 induced decrease in lifespan. So I have pretty confidently eliminated A, B, and C, but I will still review D. So again, D says that I have an HST3 induced decrease in lifespan. Okay, that is correct. I do have an HST3 induced decrease in lifespan. And then it says when I delete the RTT gene, this has no effect on that decrease in lifespan. Is that correct? Yes, it is, because when I delete RTT, as I've done in this white triangle, that has no effect. It's exactly the same as when I only deleted HST3. So that extra deletion of RTT has no effect because they look the same, they're the same curve. So I can confidently conclude that it is D. The next question asks, what is the best experimental method to analyze the effect of the TDH2 gene deletion on the rate of histone acetylation? Comparing histone acetylation in wild type and TDH2 cells by, this is Western blot A, because that is how you would determine if histones are acetylated or not. And remember the acronym SNOW-DROP if you haven't heard of it. You can Google it. It's a really helpful technique in remembering what each of these types of techniques help us visualize. So RT-PCR, that is for amplifying genes. Southern blot is for DNA, and northern blot is for RNA, whereas western blot is for proteins. Next question. Which experimental approach can be used to analyze the effect of ROS on the lifespan of yeast? So ROS stands for reactive oxygen species. And again, in this question, I don't really need the passage. I think that they talk about reactive oxygen species here a little bit, but really they just tell us that calorie restriction may exert its effects by reducing the level of reactive oxygen species. They don't mention much else, but I don't need more information. So number 1 says, wild type yeast versus yeast lacking antioxidant enzymes, will that help me analyze the effect of ROS? Yes, it will, because remember, antioxidant enzymes have to do with reactive oxygen species. So I think that would be a good experiment to do. It would help me compare lifespans, because it will help me analyze whether, if yeast do not have antioxidant enzymes and they cannot get rid of reactive oxygen species, what effect will that have? 2 says wild type yeast versus yeast overexpressing antioxidant enzymes. So 1 and 2 are two sides of the same coin. If I pick one, if I say that I should look at yeast lacking antioxidant enzymes, then I should also look at yeast overexpressing antioxidant enzymes. So I can't pick one and not pick 2, and I can't pick 2 and not pick 1, because they're the same. They're two sides of the same coin. There is no logic behind looking at yeast lacking an enzyme and not looking at them overexpressing that same enzyme. So right off the bat, I know it has to be D, because that is the only answer choice, which allows me to pick both 1 and 2. But just in case, I will look at 3 as well. So 3 says yeast growing in the presence or absence of hydrogen peroxide. So I know that hydrogen peroxide is a reactive oxygen species, so that will help me, that will also be a good experimental approach. And that was this passage, so it was short, but there was some good and easy analysis to be done here. And like I mentioned earlier, when I first read the passage, I don't bother really understanding everything that's going on and analyzing the figures. And as you can see, we never needed to look at figure 2, because they never asked us any questions about figure 2. So I didn't waste my time doing that. And remember, in an MCAT exam, nobody cares how well you understood the passage and the figure and the experiments. The only thing that matters is how many questions you got right. So you want to maximize the time you have to do that, and strategize your time and your questions effectively.